We're going to discuss the research article, Modulation of Taste Effect by Hunger, Caloric Satiety, and Sensory-Specific Satiety in the Rat, done by Berridge, 1991. The goal of their research was to find how hedonic and aversive reactions differ based on how hungry or how full the rats are. There were two experiments conducted. In the first experiment, they wanted to observe the hedonic and aversive reactions in rats to sweet tastes when the rats were in either a calorically deprived state or a calorically satiated state.
The goal of experiment 2 was to test sensory-specific satiety and whether the satiety from one food would result in rejection or pleasurable responses of a different food.
Before we get to the experiments, here are a few vocabulary words to help your understanding. Hedonic reactions are behaviors that are displayed towards a stimulus that is perceived as pleasurable. There are three types of hedonic reactions displayed in rats: rhythmic tongue protrusion, lateral tongue protrusion, and paw lip. Adverse reactions are behaviors that are displayed towards a stimulus that is perceived as unpleasant. And there are four types of these reactions that are seen in rats: gape, head shake, face wash, and forelimbs flail. A quick video by Berridge showing the hedonic and aversive reactions in human infants and adult rats. Satiety is when the rats are fed to or past when they are gratified. In experiment 1, caloric satiety is used, and this is when they are satiated with food that is not associated with the test substance. In experiment 2, sensory-specific satiety is used, and this is when the rats are satiated with a substance that they are later being tested with to see if they still want. Now let's discuss experiment 1. The aim of experiment 1 was to observe the hedonic and adverse reactions in rats to sweet taste when rats were in a calorically deprived state versus a satiated state. Three different types of sweetness was used: a 4% sucrose solution, a 17% sucrose solution, and a 7% sucrose with a 1% quinine solution. The 4% sucrose solution and the 17% sucrose solutions don't tend to cause adverse reactions in rats, so that's why the 7% sucrose and 1% quinine solution, which is a bitter taste, was used. The psychological conditions were that each taste was given to the rats when they were in a certain psychological state. They were either hungry or satiated. The rats' reactions were tested when they were in two types of caloric hunger conditions and two caloric satiety conditions. The two caloric hunger conditions were either 24 hours or 48 hours of food deprivation. In the two caloric satiety conditions, they were either voluntary or involuntary. The voluntary satiety was induced by allowing the rats to eat unlimited amounts of food for 1 hour for 3 successive days prior to test. In the involuntary satiety condition, it was induced by a gastric intubation, which fed them a milk and water solution without oral consumption. Alright, we have here our graphs. We have sucrose 4% is figure 2, sucrose 17% is figure 3, the sucrose quinine or bittersweet solution of figure 4, and the water, which was distilled and considered a neutral, in figure 5.
First, we're going to look at the negative anesthesia of caloric satiety. That basically means the suppression of the hedonic reactions due to satiety. All hedonic reactions were suppressed by voluntary and involuntary satiety, with the involuntary satiety being at least as strong as the voluntary, if not stronger. All but water was suppressed below the baseline, which is indicated by the solid green bars. Water was not suppressed significantly compared to the baseline, but it was significantly suppressed when compared to both levels of deprivation. Next, we'll be discussing the positive anesthesia of caloric deprivation. This is basically enhancing of the hedonic reactions due to deprivation. Overall, the hedonic reactions were enhanced above baseline by 48-hour deprivation, but not by 24-hour deprivation. The sucrose quinine solution was enhanced after 48 hours, but not after 24, but the 14% and 17% sucrose solutions were not enhanced above baseline for 48 hours or 24 hours. This indicates a possibility of what's called a positive ceiling of pleasure, where basically they liked it so much that they couldn't really go any higher. Water was not significantly elevated above the baseline, but it was higher than the satiety conditions as discussed before. The last thing that we'll be discussing with experiment 1 was aversive reactivity. This is basically the aversive reactions. These weren't affected by physiological states, whether they were full or deprived, but it was affected by taste quality. Basically, the sucrose and quinine solutions had more aversive reactions than the other, more sweeter options did, or the water. In summary, the hedonic reactions were reduced below normal levels by caloric satiety and were enhanced above normal levels by 48-hour deprivation. Suppression effect of satiety appeared to be more potent than hunger. The significant enhancement of hedonic reactions by 48-hour deprivation was missing for pure sucrose taste, the 4% and 17%, suggesting caloric hunger may not be able to enhance palatability beyond a certain level. These data support the idea that hedonic taste palatability is evaluated separately from taste aversion. They also indicate that hunger and caloric satiety both relate directly to the hedonic dimension of effective processing, and shift hedonic perception of the taste without altering the aversiveness of that taste. This is important because an arousal factor would be expected to alter all responses, aversive and hedonic, not simply the hedonic ones as indicated here. Is going to be discussing experiment 2.
In experiment 2, there were 11 female rats. The rats were free to eat and drink water and pellets and milk and sucrose for 1 week as they pleased. In the sensory-specific satiety method, the rats were orally infused with either milk or sucrose until the rat rejected it either actively through aversive reaction or passively through fluid drip out of mouth. Once they rejected it, they waited for 30 seconds and then pumped again. If the rat rejected within 15 seconds, the rat was considered sated. In the taste reactivity method, 1 minute after rejection from the above method, a new tube of either the sated food or the other food was given for 30 seconds. Baseline with the milk and the sucrose was tested prior to testing, and both of these methods were only done 1 time each day. The results of this experiment 2 were, by making the rats full, it reduced pleasurable reactions to the milk or sucrose, regardless of which one they got full from. Aversive reactions were not increased by caloric or sensory-specific satiety. These findings replicate the suppression of hedonic reactions by caloric satiety found in experiment 1. Additionally, they show that hedonic suppression may be strengthened further by sensory-specific satiety to food. Sensory-specific satiety reduced the hedonic palatability more than just caloric satiety. Both reduce the hedonic properties without altering taste aversion.
Basically, the takeaway from this study is that the changes in food affect produced by hunger and satiety are restricted to the hedonic dimension and do not actually affect the aversive dimension.